Okay, so I have a small correction to make. This is on slide 1910 from the last lecture. I had on the slide the word same tRNA, but it should really be a different tRNA. It has a slightly different sequence for elongation methionine than for initiator methionine. So if you just cross that same and write different, then we'll all be on the same page. The right page. Okay, today we're going to finish up the molecular biology section by doing recombinant DNA and cloning. So what is recombinant DNA? Recombinant DNA is DNA that has been combined from two different sources. And frequently these different sources are different species. And a word that I've added here that I don't think is on your version is in vitro. So the key part here is that this is done in the lab. It's not just combining DNA through selective breeding, through standard breeding approaches, but we actually take the DNA and manipulate it in the lab. And this lets us do things that are impossible through standard breeding approaches, right? Last time I showed you those fluorescent green mice. Jellyfish and mice cannot mate, so the only way to produce a green fluorescent mouse is using recombinant DNA technology. Now the way we begin doing recombinant DNA technology is by cloning our gene. So what does that really mean to clone a gene? Most people mean that you've taken your gene sequence and put it into a form that can be manipulated in the lab. So generally that would be putting your gene into a plasmid. And we're going to start by talking about what some of these basic tools are for recombinant DNA technology. The first one is a plasmid that we just mentioned. Probably you guys have heard about these before. They're small circular DNA molecules that replicate separately from the bacterial chromosome. So the bacterial chromosome is also circular DNA, but the plasmid is a separate piece of circular DNA that's a part or separate from the bacterial chromosome. Plasmids are useful for storing, amplifying, or expressing DNA. We'll go over those things a little bit more on the next few slides. These plasmids are propagated in bacteria, and bacteria are great tools for recombinant DNA research because they grow very quickly. E. coli divide every 20 minutes under optimal conditions. And they grow pretty cheap. They don't need expensive food. So they're a lot easier to culture than mammalian cells. So when we can, we use bacteria rather than mammalian cells to produce proteins. Restriction enzymes are proteins that they're enzymes that cut phosphodiester bonds on both strands of double-stranded DNA. We use these guys to move DNA around. We'll talk about that some more today. One thing I wanted to point out is these are also called restriction endonucleases. And the endo here means that they can cut in the middle of a piece of DNA. As you go through your biology curriculum, you'll encounter other nucleases that are exonucleases. That means they can only remove the last nucleotide. They can only cut the last phosphodiester bond. These guys are endonucleases, so they can cut right in the middle of a piece of DNA. Makes them a little bit special. Now these plasmids come from bacteria in the wild. Plasmids are a natural thing that we have taken from the bacteria and modified to make them very good and useful for carrying our genes around. So the origin of plasmids was wild bacteria. And now the ones that we use in the lab are modified versions of those original plasmids. We use these plasmids to carry our genes of interest that we can get from a variety of sources. Here this looks like a mammalian cell, and we're getting some genomic DNA out of it. And we need to get this gene into the plasmid. There are a few different ways you could do this. One way would be PCR, the polymerase chain reaction. We're not going to talk about that in 593, but you'll certainly hear about it in future classes. What your book talks about is cutting this genomic DNA with restriction enzymes. So the restriction enzymes will cut it into little pieces, and then these pieces can be moved into the plasmid. Once your gene of interest is moved into the plasmid and you put it back in some bacteria, oftentimes you'll say you cloned your gene. Now these plasmids that we use in the lab have a couple of important properties that I want to go over. Of course, they're circular. We already talked about that. And another one is that they're small. So KB stands for kilobase. That's 1,000 base pairs of DNA is a kilobase. So these guys run from about 1,000 to 10,000 base pairs of DNA around the circle. It's kind of an average size. They're not big, like the mammalian genome is 3 billion base pairs. They have an origin of replication. It's often abbreviated ORI. And that's important because they're separate from the bacterial chromosome. So if you want the plasmid to be replicated and passed on to the daughter bacteria, then they need to have their own origin of replication. They also have a selection gene. Often these are antibiotic resistance genes. The one shown here is the ampicillin resistance gene. This is a gene that produces the protein beta-lactamase. Beta-lactamase is an enzyme that inactivates antibiotics in the penicillin family. So ampicillin is related to penicillin, and it's inactivated by this enzyme beta-lactamase. Now this selection cassette here is important because it allows you to kill off all the bacteria in the culture that don't carry your plasmid. Only bacteria that have this plasmid will make the protein that allows them to be resistant to ampicillin. So that's a key part of the plasmid. Over here in green, we have a region where we can put our gene of interest. And this can have lots of different things in it besides just our gene of interest, but it generally contains the restriction sites that we're going to use for cloning. It may contain other genes we want to use for selection purposes, and sometimes it'll contain a promoter. So if you want to express your gene of interest, of course you're going to need a promoter. If you want to express your gene of interest in bacteria, you'll need a bacterial promoter that the bacterial transcriptional machinery will recognize. If you want to express this gene in mammalian cells, you'll need a mammalian promoter. And this is something that people often get tripped up about. There are plasmids that you can use to express genes in mammalian cells. We have ways of getting plasmids into mammalian cells, and we can use the plasmids to express mammalian genes in mammalian cells. Now, if we want to grow up lots of this plasmid to propagate it, we do that in bacteria, but we can also use plasmids to express genes in mammalian cells. Okay, so we cloned our gene into the plasmid, and we grew up a bunch of bacteria carrying that gene. We have two directions we can go from there. One is to study the gene, and one is to study the protein. So ways you might study the gene or use the gene would include making genetically modified organisms, or GMOs. One example of that is there are crops that pesticide resistance genes, or genes that kill pests have been introduced into them, like a Bt gene allows corn and soybeans to be resistant to certain bugs that want to eat them. You may have heard of the Deepwater Horizon oil spill, that they were using bacteria that could digest hydrocarbons, and some of those have been engineered in the lab to express the genes we want them to have. We talked earlier about how you could make proteins like insulin, human insulin in bacteria. You can make other human proteins that have therapeutic value as well. These would be called recombinant proteins because we produce them using recombinant DNA technology. We combine DNA from two different sources to make them, and they are made in the lab. This question is just for fun, so just take your best guess. Which of the following is not a real GMO? Find the imposter. Okay, give it your best guess. See what you guys like. Okay, the popular imposters were the watermelon and the spider goat. I thought those were improbable. Okay, well, these are the real GMOs. So they have made supersized salmon that grow twice as fast as regular salmon. They've made environmentally sensitive pigs that have less phosphorus in their waste, so that doesn't poison the environment as much. Spidey goat here makes silk milk. He secretes, well, she puts spider silk into her milk, and you can purify that and make biosteel. Pretty cool, huh? And this is a genetically modified cat that lacks the main feline antigen that people are allergic to. So there's one protein that causes most of the problems, and there's a couple companies making cats that lack this gene. They've removed it. This cat was actually produced by selective breeding, so it's a little bit cheating, but there are people making knockout cats. This was the imposter. These guys are just born in a box. So that's kind of sad, but it's true. Look at that. $100 for your square one. Yeah. If biology doesn't work out, we can always grow square one. Okay, back on track. We're going to talk about restriction enzymes. So how do you get your gene into that plasmid? He said one of the ways you could do it was using enzymes that cut the DNA into little pieces. These are restriction enzymes. They're restricted to cutting a certain sequence. That's not exactly where their name came from, but you can remember it that way. So restriction enzymes tend to cut palindromic sequences. Not always, but much of the time, they cut palindromes. So what is a palindrome? Probably the most famous palindrome is shown here. This was written by Napoleon in his cell on the Isle of Elba. He was imprisoned after he lost. And what he wrote was, Able was I, ere I saw Elba. And you see, if you read that backwards, you see, Able was I, ere I saw Elba. So a palindrome is something that reads the same forwards and backwards. So let's see what that means in terms of DNA. Remember we talked about the Tata box, part of the promoter, and we said that's not palindromic. When the RNA polymerase was up here, it looked at it this way, it looked at it this way, it did not look the same, right? You flip this around, and it doesn't say TATAAA anymore. It's something different. It's not palindromic. Now, EcoR1 is a famous restriction enzyme, and its site is a palindrome, GAATTC. Now this site is double-stranded DNA, so this is one way to write it. You have GAATTC, and when you flip it around, you actually still end up with GAATTC. So it's palindromic. It reads the same in both directions. No matter which side of the DNA you're standing on, that restriction recognition site is going to look the same. I added the word can here. Restriction enzymes often, some of the most useful ones, make staggered cuts. They produce what we call sticky ends or overhangs. One example of this is our friend FOR1. Here's its recognition site, GAATTC, double-stranded DNA. Now, when this enzyme sees this site, it will make a cut in the phosphodiester backbone of the DNA. It'll break this phosphodiester bond between G and A. So it'll be left with the hydrogen bonds. It'll make a corresponding cut on the other strand between the G and A. So it cuts in the same relative location on both strands, but they're staggered because the recognition site is 6 nucleotides long. Now these hydrogen bonds, are not stable enough to hold the DNA together without the backbone. So eventually these will drift apart. We often draw this recognition site as just 6 nucleotides, so I wanted to make sure you guys thought about it in this way. Oftentimes, these sites are in the middle of a piece of DNA, so there's lots of DNA on either side of these sticky ends. So those are the pieces that we might want to move around. Each restriction enzyme is going to have a slightly different recognition site. There's another famous enzyme called BAMH1. And its recognition site is a little bit different than EcoR1. Instead of seeing GAATTC, BAM sees GGATTC. So it's still a palindrome, but a slightly different sequence. It cuts in a very similar way, but it will cut between these two Gs, and then again, between these two Gs here. And with just the hydrogen bonds holding it together, eventually these guys will drift apart. And just as before, these sticky ends can be on the ends of a big piece of DNA because these are endonucleases that can cut in the middle. Now there are such things as blunt cutters, enzymes that cut in the same place on both sides, both backbones. So there will be no overhang. If I cut this bond here and this bond here, that would produce a blunt end with no overhang. Those enzymes are used sometimes in cloning, but they're a little bit trickier to work with. So the guys that produce sticky ends are the most popular. Now, having these sticky ends allows you to put pieces of DNA together that have the same sticky ends. So if I cut the black piece of DNA with echo R1, and I cut the gray piece of DNA with echo R1, I can use molecular biology techniques to get rid of those two, and then I can try and put these two together. I can try and get them to anneal. So in this context, the term anneal refers to two pieces of complementary DNA forming hydrogen bonds. So annealing means that two pieces of complementary DNA are forming hydrogen bonds. And these guys will eventually drift together and form hydrogen bonds because they are complementary. Their sequence matches exactly. Now if I cut the black piece of DNA and the gray piece of DNA with two different enzymes, then they wouldn't do that. So again, I'm going to try and put these two fragments together, but now they have different sticky ends. Their overhangs don't match. So they wouldn't anneal because A will not base pair with C, and over here G will not base pair with T. So it allows you to put ends together that have complementary overhangs specifically. Okay, we're going to try a demonstration. Hopefully it will help you remember some aspects of this. I'll need 4 volunteers. 1, 2, 3, 1 more. Wave your hand if you have that because I can see it. Okay, excellent. Okay, first one down here, you get to be echo R1, cheat sheet. Okay, you can be BAM, got a sign in the cheat sheet. I'll give you ligase. Okay, you've got some clips on there, remember those later. There you go, you are ATP and you are the keeper of the high energy phosphates. Use them wisely. Okay, so echo R1, where'd you go? You, at the scissors, you are the restriction enzyme. And I'd like you to go look at our piece of DNA that we're going to clone here and see if you can find your recognition site. You guys have to stand back for a sec. I know you guys can't see this well, and I'm echoing, but she is scanning that piece of DNA, looking for her specific restriction site, which I think she has found. And now she's going to cut between the G and the A nucleotide. Very good. She's going to make a staggered cut. Only in the place where she sees her space. Okay, excellent, well done. You may pass those scissors over to BAM. And we'll see if BAM, H1, can find his recognition site in this piece of DNA. While BAM is hunting that down, I'm going to go ahead and put down my plasmid. I know it'll be a little hard to see from the back, but the people in the front will be able to see it pretty clearly, I hope. How you doing, Van? Got it? Okay, excellent. It's hard to do these things under pressure in front of 500 people, so these enzymes are doing a very good job with their specific cutting. So here, I have my plasmid that I prepared ahead of time, so we didn't have to watch them cut more DNA. And this, of course, is the origin of replication, because we want this to be propagated when the bacteria divide, and the antibiotic resistance cassette. And I cut it with the same 2 enzymes, BAM and ECOR1. That's perfect. You did a good job. I wasn't even watching. Okay, so now you 2 enzymes, ECOR1 and BAM, take off your signs. And I want you to take them off because your job is done. So I want everybody, just stay up here. I want everybody to remember that the enzymes cut, and then they're done. They go away. No more jobs for them. But the Bio-93 students who are up here now get to take this piece of DNA, insert, and pop it into my plasmid. Can you guys anneal that piece of DNA? Okay. So the Bio-93 students are smart. Did you see what they did? They turned it around, right? Because they saw that the ends wouldn't anneal the way I laid it on the table. They were not compatible. So now they're hydrogen bonded, thank you guys, which is good, but the situation is unstable. That piece will pop in and out of the plasmid because the backbone is not complete. So who do we need? Ligase, all right. So ligase, you can go make some phosphodiester bonds with your clips, but what does that cost you? What part of ATP? High energy phosphates, how many? 2 per bond. All right, you may bounce some ATP so she can clip. Yes, nice. Okay. Last person didn't enjoy the bouncing. Maybe that's the best part. Okay. You only have to do 2 in the interest of time. So she would form phosphodiester bonds on each end. Did you bounce 4 balls? Yep, 2 each. 2 for each bond. So each phosphodiester bond costs a pyrophosphate. Yeah, because she already clipped it. Okay, excellent work. All right, thank you guys. That's beautifully done. You can leave your stuff and get your treats. Thank you very much. You can pop the ball in there. So one thing I wanted you to understand is I prepared this vector ahead of time with the BAM and EchoR1 sites already cut. And when they saw that their piece was in the wrong orientation, they did just the right thing, flipped it around, and cloned it in, annealed it with my vector. And one of the reasons why I wanted you to see that is to realize that these pieces of DNA are floating around free in solution. They can move around like that. My plasmid is exactly a circle because real DNA doesn't have to stay in a circle. It can flop around. So that's important. And the enzymes were not involved in putting that piece of DNA into the plasmid. They did the cutting and then they were done. So these are some of the points I want you guys to remember from this demonstration when you're thinking about problems. That restriction enzymes only cut when they found their palindromic recognition site. So both EchoR1 and BAM knew exactly where to cut. They didn't try and cut each other's sites. They knew this is my spot and I will cut here, and they cut it in a very specific way. So the different enzymes have different recognition sites. When they tried to anneal the piece of DNA into my plasmid, they saw the ends were not complementary. In this case, they were able to flip it around and then it fit. So only pieces, sticky ends that will hydrogen bond will anneal. And last, we needed DNA ligase to form those phosphodiester bonds, our old friend back from DNA replication. And to do that, cost a pyrophosphate off of ATP each time ligase formed a phosphodiester bond. So they did a great job. Thank you for help with the demonstration. Any questions about that? You guys are all writing. This will be up in the podcast if you don't have time to get it all down. So now we're going to talk about cDNA, which is another really cool and important concept in recombinant DNA technology. The C here is for complementary DNA, and it is a double-stranded DNA copy of the mRNA that's present in a cell at a particular moment in time. So the first thing you do is you isolate the mRNA from the cell. Then we're going to copy it with a new enzyme that we haven't talked about in this class, reverse transcriptase. It's got that ACE on the end, so you think it's an enzyme, which it is a protein, again. This is going to do reverse transcription. So now this is going to convert RNA into DNA, the reverse of normal transcription. This is a DNA polymerase, so it can't start from scratch. It's going to need a primer. And we prepare this primer in the lab. We make a string of thymidine residues, oligo-DT. And we make the string of T's because the end of every mRNA is what? A string of A's, right? So these T's will base pair, anneal, to every mRNA. So those are great primers for this first step. When we do that, we will synthesize the DNA copy of the original mRNA. Now we're trying to make double-stranded DNA, so we want to get rid of this RNA part. There are enzymes that will selectively degrade the RNA. We can use those to get rid of the RNA. There's chemical treatments we can use to get rid of the RNA. We get rid of the RNA, and we use a DNA polymerase to make that second strand. So this will be like the sense strand. It'll be the mRNA with T's instead of U's. And this DNA polymerase will, of course, need a primer too. And we have to use some special tricks to make primers for this end, but there are ways to do it. And at the end, you'll end up with a double-stranded piece of DNA that's an exact copy of the mRNA. a DNA copy of the RNA. Now, why would you want to go to all this trouble to make cDNA? I'm going to give you a couple of reasons. They're very useful. One reason is cDNA are useful for manipulating or expressing the gene. So you could inject mRNA into a cell, people do this all the time, and get the gene expressed. But RNA is not very stable. And that's not because of the chemical properties of RNA, but because there are enzymes all over the place on their skin in the environment that degrade mRNA. For us, it's part of our antiviral response to have lots of enzymes that can degrade RNA. So in the lab, we would much rather work with DNA than RNA. That's one reason why they're useful. If you think about it, when you isolate these mRNA from a cell, they are telling you what the gene expression pattern is of that cell under the conditions of your experiment. If you look at a library or a collection of all these cDNAs from the cell, you can tell what genes were on when you collected that mRNA. Then you can mess with the cell or mess with its environment and see how the cDNA, the gene expression pattern, changes. So that's another way making cDNAs can be useful. Now, I think you guys understand what the difference between a DNA and an mRNA is. And a cDNA is a copy of an mRNA. So what's going to be different between a genomic DNA gene and a cDNA of that gene? What's missing in the cDNA? The introns. That's right. Very good. Because the introns were spliced out when it turned into the mature mRNA. So a cDNA won't have introns. And again, in the lab, that's a nice thing, because introns can be very long. They can make our DNA extremely long. So having a cDNA without these introns is good, because our DNA fragment will be shorter and easier to work with. So that's helpful. Another thing that will be different if you get rid of the introns is you won't be able to alternatively splice that gene anymore. So if you want to study a particular splice isoform, a particular form of that protein, you won't get splicing anymore if the introns are gone. And if you want to study splicing, the cDNA may be a little bit trickier to use, but alternative splicing will be gone. No need for splicing. Another reason why it's good to get rid of those introns is, remember, we talked about expressing insulin in bacteria and using that to make a recombinant protein for diabetics to use. But bacteria, you may recall from your reading, don't have a nucleus. They couple transcription and translation. They don't have a spliceosome, so they can't splice out mammalian or eukaryotic introns. They cannot deal with introns. If you want bacteria to produce your gene, you have to get rid of the introns. You have to give them a cDNA as a template. Okay, see if you guys got this. A cDNA would include which of the following sequences? Okay, go ahead and choose your favorite. All right, let's see what you guys liked. All right, so you guys got the intron point. That's very good. But you're kind of split between the other three possible answers. So let's have a look at it. The promoter. I'm going to go back here. So here's our promoter. The top box is probably here. There's some other elements here where the specific transcription factors bind. And the transcription unit starts here. So is the promoter likely to be in the cDNA? No. There might be some small part of the promoter in there that's downstream of the transcription start site. But for the most part, you will not find the promoter sequence in the mRNA or therefore the cDNA. So the promoter is out. What about the 3' UTR? Would that be present in the mRNA? Yes. Remember, that's exon sequence that doesn't code for protein. So all the exon sequence is present in the cDNA. So yes on the 3' UTR. This is the right answer. The 5' cap. I realized that I don't think I showed you guys a picture of what the 5' cap looks like. You don't have this, but it's on Wikipedia if you wanted to look at it. So the 5' cap is a modified guanosine, but it's not attached with a phosphodiester bond. It's a strange phosphate-phosphate bond. So that's not going to get copied by a DNA polymerase. So the 5' cap is not correct. The correct answer is the 3' UCR. Does anybody have any questions about that? Good. Okay, so now we're going to talk a little bit about what I think the general public thinks of when we say cloning, and that's cloning an organism. We're going to start by talking about somatic cell nuclear transfer, which is the way the sheep dolly was created, if you've heard about her. Now, the way this started is they took some mammary cells. It doesn't really matter that they're mammary gland cells. It's just what they happen to use. And they cultured them under starvation conditions. And they did that because it turned out to be important to get these differentiated cells out of the cell cycle in order to be able to do this technique. So they start out by starving those cells. Over here, they took an egg or an oocyte from a different sheep. It's a black-faced sheep instead of a white-faced sheep. And they took the nucleus out of this oocyte. So now you just have egg cytosol with no nuclear genetic material. Then they took the mammary gland cells that had been starved, and they fused them with this enucleated egg. They did this by giving them an electric pulse that caused the membranes to fuse, and you end up with the nucleus from this differentiated somatic cell floating in the egg cytosol. And these two things together, the starving the differentiated cell plus bathing it in egg cytosol, were sufficient to reprogram that cell into an embryonic state, so that now it divided to make an embryo. And that was using just the genetic information that was in that differentiated mammary gland cell. They implanted that embryo into a surrogate black-faced sheep and got out the white-faced sheep, just like the mammary cell donor, and they named that sheep Dolly. This is something I added to my slide, so I wanted to make sure that you got the text of this right. So one of the reasons why this experiment was so important is it showed that there is no irreversible modification of the genetic information in an adult differentiated cell. We've been talking a lot about how every somatic cell in your body has all the information theoretically necessary to make a new you. But it wasn't clear whether that information had been changed in some way, that it was no longer competent or able to make a new person or sheep. But this experiment showed that there is, in fact, all the information you need to build a new organism present in the somatic cell nucleus. So if you can make a sheep this way, can you make a human this way? Yeah, in principle, you certainly could. But most scientists really don't want to do that. This is the stuff of science fiction, right? Like in the Clone Wars, where they made a bunch of, what's his name? Oh, I'm getting old. Fett. Jango Fett. Where they made a whole army of Jango Fetts and now save the day and all that. But real scientists don't really want to clone humans, except in a special way. So when would you want to clone a human? That would be to make parts, not to make people. This is called therapeutic cloning. This is an example of how it would work. So if you take some embryonic stem cells and you culture them under the right conditions, you can get them to differentiate into a whole bunch of different kinds of cells. So you could take embryonic stem cells and take them down the path of becoming neurons. Then if you have a spinal cord injury and need to make some new neurons, you could inject these cells, and maybe they would form the right connections and maybe restore some of your function. So that would be one application of stem cells. There are many. Problem is, if you use the existing human embryonic stem cell lines to make these neurons, they would be recognized as foreign by your body. They're not your own tissue. It would be just like getting a lung transplant or a liver transplant. You would have to take immunosuppressive medicines for the rest of your life, or your immune system would destroy those new cells that you got. But if you use somatic cell nuclear transfer, you could take a skin cell from me, take its nucleus, pop it in a human egg, get it to differentiate, get it to form a blastula, take the embryonic stem cells, differentiate those into nerve cells, and now we've made nerve cells from my own DNA. They will look exactly like me. So you can make an autologous transplant that won't be rejected, and you won't have any need for immunosuppressive therapy. So that would be a really useful way to use this technique. Now, something that's been in the news recently that's even cooler is the generation of iPS cells, or induced pluripotent stem cells. Induced means that they were made to change. In this case, a somatic differentiated cell was made to change into an embryonic cell. They're pluripotent. This means they can differentiate into a variety of different cell types. And they're stem cells. Stem cells can replicate indefinitely, and under the right conditions, they can be induced to differentiate into a variety of tissues. So here's the basic protocol for how you make an iPS cell. You start, just like in the somatic cell nuclear transfer experiment, you start with an adult differentiated cell. Now they used a mammary gland cell in that sheet, but you wouldn't have to do that. You could use any somatic cell. It's just skin is easy to get, so people start with skin. They isolate those cells and grow them, and then instead of using an oocyte to reprogram and starvation, we induce reprogramming in the original experiments with 4 transcription factors. So remember that transcription factors are key regulators of gene expression programs. So by putting these 4 transcription factors into these cells, you can reprogram their gene expression patterns to be embryonic. You culture these guys for a while, and then can put them under conditions that can make all sorts of different kinds of tissues. So this is even cooler than the last one, because now I don't even need a human egg. All I need is some cells from my skin, or a cheek swab, or some blood. Any nucleated somatic cell will do. Now, I really wanted to show you this video, but there's not going to be time. So I really want you guys to watch this at home. I was going to show you the last half, but it's not that long, so you can watch the beginning of it too if you like. This is a link. But I'm going to summarize what you will see in that video when you go watch it here. And there's a link to the original paper if you want to read it. It's not hard reading at all. So this particular group started out with a mouse that was a model for sickle cell anemia. They had a mouse that had the same mutation that humans with sickle cell anemia have. And these mice had the same disease that people have. They took some skin cells from that mouse, grew them in culture, and added the reprogramming factors. So those 4 transcription factors were added and induced those cells to become embryonic stem cells. Now these embryonic stem cells still have the sickle cell mutation. So what they did was they fixed it. Embryonic stem cells are special because you can switch pieces of DNA around. It's called homologous recombination. So they were able to fix the sickle cell anemia mutation, repair it. After the cells were fixed, they induced them to differentiate into blood stem cells that could make red cells and other cell types. They injected these into the sick sickle cell mice and cured them of the disease forever. Because those stem cells will go into that mouse bone marrow, set up shop, and live there. These were the mouse's own cells, so they weren't recognized as foreign. They weren't rejected. It's a very cool experiment, and I think it gives great hope for new ways we might treat sickle cell disease. The work that led up to this experiment was performed by a couple people. John Burton's experiments are in your textbook. You can read about them. And Shinya Yamanaka is shown in that video that I posted the link to. So the key with the iPS cells is now you don't need a human egg cell to make the embryonic stem cells. So that's a key advance. There's a couple points that I want to make sure you guys understand about iPS cells. First is that this reprogramming comes from changing the gene expression patterns. So the adult somatic cell goes back to embryonic. This is done by adding transcription factors. They are key regulators of the gene expression program. The way they did this in the lab was using recombinant DNA. But there's some problems with that. Those viruses you've used can actually cause cancer on their own. So people are trying to find chemicals that they can use that will have the same effect as these genes. That might be safer. And this is something everybody should understand about stem cell therapies, as there are so many being popularized in the public literature now, is that you have to be careful. Stem cells have many properties in common with a cancer cell. They proliferate indefinitely, they're dedifferentiated. A lot of times these stem cells can go on to produce tumors. So you can give somebody cancer when they're curing their original disease. So we have to find ways to stop this from being a problem. Okay, on Friday, we're going to talk about Mendel. I finished a few minutes early, so come ask me questions out here if you want. See you Friday.